Hello everyone. Today we are going to continue, and this will be the last session for this theory session for this biotechnology and its application. After this, from next class we will solve its NCERT exercise and NCERT exemplar. In theory, it is the last session, that is NCERT session 10.3 and 10.4 of this chapter, which is biotechnology and its application. So let's get started. Disclaimer. So, what are going to be today's learning objective? So, first of all, we're gonna define what is transgenic animals. Then, we will identify the reasons for producing transgenic animals. That why is there the need to produce this transgenic animals? What benefit they are causing to the society, to the humankind? That's why we are producing this transgenic animals. So, we need to know the reason for that. Then we will recognize the ethical issues related to genetic modification. We can't, we can't use this, we can't alter every genetic of, we can't disturb the ecological system. We could not change the genome of every individual. We could not make this whole world transgenic. Is some ethical issues that is related to this genetic modification, and that we are going to see today in today's class. Then we will explain what is biopiracy. Then we will understand that what are the different measures that has been taken against this biopiracy. So this is the whole objectives that we are gonna learn in today's class. First, we will see the transgenic animals, why we produce them, what are the ethical issues that we cannot convert every animal, every human into transgenic animals and all. And then we will see what is biopiracy, and that we will see what the government has taken measures against this biopiracy. So what is going to be today's index? So first we will cover this transgenic animal, which is the 10.3 NCERT session. Then we will study about the ethical issues. Then there will be a summary where we will see the gist of all classes. Then there will be some NCERT based questions which we're gonna solve together. Further, there will be some keynotes, and then we will conclude today's class with some interesting questions. This slide with some interesting questions. So now comes the topic, what is transgenic animals? Okay, what is this definition of this transgenic animals? Animal that have had their DNA manipulated to possess and express an extra or foreign gene. So what is a transgenic animal? So transgenic animals are the animals whose DNA has been altered so that they can express a gene. Here that gene termed as foreign or extra genes. So what is that transgenic animals means? Animals that have their DNA manipulated, means that have their DNA altered so that they can possess, that they can have an extra gene or an foreign gene, and they can show that particular trait of that gene. So that is what we call transgenic animals. So what are the example of these transgenic animals? Rat, rabbits, pig, sheep, cows and fish has also been produced. So the DNA of these animals have been manipulated, has been altered so that they can express, they can show some foreign trait, some foreign gene. That foreign gene will show some trait only. That's why I'm saying that they are showing some foreign trait, which is not their own trait, but they are showing because of the presence of that extra foreign gene. So till now we have able to make this make these animals, that is rats, rabbits, pigs, sheep, cows and fish, to transgenic animal, that we are able to alter their their genetic material, that is their DNA. In today's class, we will even see some examples that how these animals DNA have been manipulated to show some extra trait. So, over 95% of all existing transgenic animals are mice. So, most of the experiments have been done to on mice to make it transgenic. So, among all these examples that we have given, 95% of the existing transgenic animals are mice. Others consist of among the 5 other percent. So here you guys can see a transgenic mice. They have made this mice transgenic in a laboratory. So this is one of transgenic mice. Next. So as we know that what are the transgenic animals? The animals that have that have a manipulated DNA so that they can have an extra or a foreign gene, and they can show an extra trait, a different trait. But why is there any necessity of producing this transgenic animals? They are doing their own work. Okay, they are in this ecological system. They're doing their own work. They are eating their food and maintaining all the ecological balance and everything. Then why there is need for this to make them transgenic animals? Why we need to change their genetic material, alter their genetic materials? So here are some reasons that makes it necessary to make transgenic animals. So what are those necessities? What are those factors? So what are those things for which we produce these transgenic animals? So first is to study normal physiology and development. So what do you mean by to study normal physiology and development? What is the purpose of that? To study how genes are regulated. Okay, so as we know, like a like a human have lot of genes. Okay, and every gene has their own function. They produce a particular protein, and that protein has their own function and all. So how we will understand what this gene was doing in our body? What was its role in physiology? What was its role in development? So we can study that thing using these transgenic animals. We can study that how these genes are regulated by introducing these genes in those animals, and then we will see, okay, what trait they are showing, or what reactions happening in their body, and all those things, how these genes are regulated. So these kind of study we can do with the help of this transgenic animals to understand how they affect the normal function of the body and its development. So, as I said, for example, there is a gene in a human, okay, and that is responsible for some development and some normal functioning of the body. How we as a people will get to know that, okay, what is the function of that gene? So, what we can do, we can introduce that gene in that animal, make it a transgenic animal, and then we can study, okay, so this gene is responsible for this functions, or okay, this gene has been regulated like this. These are the different physiological functions happening in the body after we have introduced that gene in the animal. So, to study this normal physiology and development, we think that there is a necessity for this transgenic animal. Then other reasons. So, okay, so before other reasons, let's see the example. So, as I said about, like, we need to see how that gene has been regulated and all those things. So, there is an example that studying complex growth factors such as insulin like growth factor. Okay, so our body do have a complex growth factor, which is we call insulin like growth factor, IGF. It involves introducing genes from other species to alter its formation. It allows us to observe the biological effect of these genetic changes. These observations help gather information about the factor's role in the body. This approach provides insight into the biological functions and significance of IGF. So, What this example is telling us, it is telling us that, see, there is a growth factor in a body, okay, and it's a very complex growth factor which we call insulin like growth factor, IGF. So now what it happens, it involves introducing genes from other species to alter its formation. So to alter its formation they need genes from other species, okay. So now what we can do with this observation, what can we like gather information about that? So this observation help us to gather information about the factors role in the body. By this we can see what is the factor of the, what is the this factor roles in the body. And this approach, what will help in, by knowing this, what will help us to like, what we get to know, we will get to know that what is the biological function and significance of that gene, okay. So this is one of the example. Then after that, to know and to understand this normal physiology and normal function of the development of the body, we can also study about the disease. How we can study about this disease, what is the purpose of that? Transgenic animals serve as a specific model for human disease. For example, if we know that a particular disease happening because of the this gene, okay, we can introduce that gene into that body. Or if that we know that, okay, this gene getting mutated, and because of the mutation of this gene, this disease is happening to a human. So what we can do, we can also use that mutated, insert that mutated gene into that transgenic and into that animal to make it a transgenic animal. So now what will it do? So now that animal, that transgenic animal, will serve as a model for that human disease. So because now that gene is also, inserted into that animal, is in that animal, that animal will also act as a model for that human disease. They are designed to increase our understanding of how genes contribute to the development of disease. So they are designed so that we can understand that how those genes contribute to the development of that disease. It makes possible for the investigation of the new treatment. So what is like, why we need to have this model of human disease? So that we can find some therapeutic intervention, so that we can find some treatment, okay, against that disease. So there are examples. So examples involve current transgenic animal models exist for cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer disease. So now we have this thing that we can make this transgenic animal models for which diseases. We can make it for cancer, we can make it for cystic fibrosis, we can make it for rheumatoid arthritis, and we can even make it for this, uh, we can make it for Alzheimer disease, okay. This is a neurological disease. So for example, this cystic fibrosis. What is cystic fibrosis? In cystic fibrosis, what happen, it affects the cells that produce mucus, sweat, and digestive tissue juices. And this, the cells that are producing mucus and sweat and all those things, so this have started producing that in very large amount. And what happened, so we can, the body start producing abnormally thick and sticky fluid, okay. And this happened because of a defective gene in our body. So now if we have to study this cystic fibrosis in a human in a mice model, or in some, in different animal model, transgenic model, usually it is mice only. So what we can do, we can use that gene, okay, we can use that defective gene. So we can, there are some methods. So there are some methods, so we can make that mice into that particular transgenic animal by mutating their gene or by inserting any gene. There are several methods. So now it will act as a cystic fibrosis. For cancer also, cancer, we also know that there are many genes which are involved in cancer, like BRCA, TNF alpha, and all those things. So we can also make that possible. So these, using by altering this gene, or by inserting this gene, or by mutating those gene in that mice, in that transgenic animal model, we can use it as model for human diseases. Now Oh, okay. So we have till now studied that we can use this transgenic animal to understand the normal physiology and development and to study about the disease. What all other reasons that we need this transgenic animals? So we can need them to because we need the biological products from them. What is the purpose of that? Certain human diseases need medicines for treatment. We know that human diseases need medicine for treatment. Those medicines can contain biological products. These biological products are often expensive to make. As an alternative, transgenic animal could be used to produce such products. So we need know that to treat the human disease, we need some medicines. Okay. So and those medicines can contain some biological product, but making this biological product are quite expensive. So what is the alternative to that? We can use the transgenic animal to produce such products. Next we will see the example how we can use this transgenic animal to produce a product, biological product. So for example, human protein alpha antitrypsin. So this is a protein which is alpha 1 antitrypsin. So it is used to treat emphysema. Emphysema is a lung condition. Okay, it is, you guys need to know this, like this is a, even you guys have studied in class 11 breathing and exchange of gases about this emphysema disease. So this is a lung disease, and we can use this human protein alpha 1 antitrypsin to treat this emphysema. So now what will happen? This transgenic animals can be created by insertion of this alpha 1 antitrypsin gene. Means we can insert this alpha 1 antitrypsin gene to a animal and make it a transgenic animal. So now what will happen? That animal is going to have this gene, and now this gene will particularly code for what? For this protein which is alpha 1 antitrypsin. So by this we can get this biological product. Okay, might be making this antitrypsin protein in, like, in laboratory and all, it is expensive. So what we did, we use a transgenic animal so that it can produce this alpha 1 antitrypsin protein, and it is used to treat emphysema. This is a lung condition. Okay, so now there is another example of this biological product, the first transgenic cow. So the first time the DNA of a cow has been altered, and this transgenic cow was named Rosie, and this was done in year 1997. So what we did to that cow? So this cow was genetically modified to produce milk enriched with a human protein. So we modified genetically modified this cow so that it will produce the milk whose milk was going to be enriched in some human protein. Which human protein? So this milk has more amount of human protein which is alpha lactalbumin. So this alpha lactalbumin is about, like, enriched in this cow's milk, and it is like 2.4 grams per liter of this protein we can get from the milk. Means 2.4 gram of this alpha lactalbumin protein is present in 1 liter of milk. So what this milk is? This milk is nutritionally a more balanced product for human babies than natural cow milk. So this milk, who has enriched amount of this alpha lactalbumin protein, is what? It is more beneficial. It is more nutritionally beneficial for human babies than a natural cow milk. So these are the transgenic cows. Figure is showing the transgenic cows. So after studying that we need this transgenic animal to study the normal physiology, and development, then to study about the disease, then we talked about the biological product, this protein which used to treat emphysema, then this Rosie, which is the first transgenic cow. What were the more reasons we need this transgenic animals? So, to evaluate the vaccine safety. So, what is the purpose of this? Testing the safety of vaccines before they are used on humans is very important. Okay, so vaccines, we know what is vaccine. So, during the COVID, everyone got to know that what is vaccine. It is like used to prevent to happen that disease in future. So, but before, because it is so before insert before giving this vaccine to human, it is very important to test that the safety of what safety of the vaccine to use on the human. Okay, so transgenic mice are being developed for this testing. Transgenic mice are being used to test the safety of the polio vaccine. Successful and reliable vaccine test could replace monkey testing. So what happened? So as I told, we need to test the safety of this vaccine, because we need to test the safety of this vaccine because we know that what is like actually vaccine is, it is mostly like attenuated live organism. Okay, or the dead live organism which has been attenuated, but that why it might happen now that you don't know that whether that vaccine is actually attenuated or not. That vaccine, the organism that has been used in that vaccine is like is still working. So we can't give it to human directly. We have to test the safety of that vaccine before. So initially we used to do it on the monkeys. So but now what happened, this transgenic mice has been used to test the safety of this vaccine. So this transgenic mice even is used to test the safety of polio vaccine, and this successful and reliable vaccine test could replace this monkey testing. So yeah, so this is also one of the reasons. Then chemical safety testing means toxicity testing. What is the purpose of this toxicity testing? To determine the toxicity and safety of drugs and other chemicals. So just like we have to check the efficacy or safety of this vaccine, we have to check the safety and toxicity level of the drugs. Like what is the safety of the drugs and these other chemicals? Okay, even the safety, and we have to see, okay, so if there is some side effects symptoms of these drugs, so what are those symptoms? Is that symptom is not too far-fetched that even that can cause some hazard to that person? Okay, that toxicity level should be checked. If it's causing normal fever or it is causing something very like heart attack, so we have to test the toxicity and even safety of drugs and other chemicals before using it on the humans. So, what are the example? Transgenic animals are created that carry genes making them more sensitive to toxic substance. So, how these transgenic animals now created for this chemical safety or toxicity testing? So, what we do, we create these transgenic animals. So, this transgenic animals carry some genes which is making them more sensitive to those toxic substance. So now if they are more sensitive to those toxic substance, so now it is we know that it is going to affect that transgenic animal. So then what we will do, we will expose those transgenic animals to those toxic substance, and then we will study the effect. So okay, then we will see, okay, so if a person, if this animal is sensitive to this toxic substance, so these are the side effects, these are the problems that is going to happen to that animal. So by that we will got to know we will able to evaluate that drug on both toxicity level and the safety level. Toxicity testing in such animals will allow us to obtain results in less time because we have made that animal sensitive to those toxic substance. Now what will happen, what is the benefit of that? That now we are going to obtain this toxicity result in very less time. Okay, we don't have to wait for too much. So till now we have studied that what is transgenic animals and why it is necessary to have the transgenic animals, and between that we have studied many examples also. Okay, so I am moving ahead. So now we need to talk about what are the ethical issues that we that should be taken care of before making this genetic modification in any animal, any human, or any further thing which we want to do. So why is there some necessity for this ethical standards in genetic modification? Why we need this ethical issues, that there should be some standard, there should be some standardized rule that everyone follow when we are talking about this genetic modification thing? Why? So first, the ethical dilemma. Genetic modification can have unforeseen ecological impacts. This line means, so we know that the earth is made such that everyone has their ecological functions. Okay, everyone has their ecological impact on earth. Okay, we have a food chain cycle, and every person, even from small to small animals and to big humans, and everyone is have some you know ecological impact. For example, if all deer will die, or all deer or every prey that this tiger feed on lion tiger feeds on die, then what will happen? Tiger also die. So there is a ecological balance in our on our earth. So if we will do this genetic modification, so it might have an unforeseen ecological impact. It can have the tendency to disturb our ecological balance. Human That's why human manipulation of living organism, so the manipulation, the alteration, or anything we are doing, or modification we are doing in this living organism, we have some regulation, we have some rules that we have to follow. Ethical standards are necessary to assess the morality of actions affecting living organism. So why this ethical standards are necessary? It is necessary so that we have some moral values, or we have some morality, so of actions, that morality of actions which is affecting this living organisms. So there should be some ethical standards. We can't do anything or everything, or we can't change whole genome of anyone's, like, we can't disturb any ecological balance. We have to do everything in a rule regulation. That then, for even in our daily life also, we have some regulations, okay, that we have to go to school at 9, come at 2, and everything. What if you guys stop going to schools? Or what if you guys just go at 9 o'clock morning in school and coming in like night or whatever? So it will disturb the balance, it will disturb the logic, it will disturb the whole balance, and whatever it is like running through, whatever the world is running through over years. But that's why there should be some rules and regulations. For like that only, making this genetic modification in human, there should be a morality of action that this ethical standards are having, and we have to use it, we have to keep it in the mind before doing some genetic modification, before making any transgenic animal. So what is the Indian government regulation? So we have an organization which we call GEAC, that is Genetic Engineering Approval Committee. What this committee is responsible for? To make decision regarding the validity of GM research. So before doing any genetically modified research, we have to take permission from this organization, and that they will say ki, okay, this research you guys can do. To ensure why they're doing this, why we have to go to them, because they will ensure the safety of introducing that GM organism for public services. It will make sure that the that genetically modified organism that have been coming, there should be safety of that, you know is it safe for the public services, it is useful and it is safe, and all those things. It will make sure that it is following all those rules and regulation, and it is not violating any rules, and that is it is safe to introduce that genetically modified organism in public for public services. So after this, what is biopiracy? Okay, so what is the definition of biopiracy? So, the unauthorized use of bioresources by multinational companies and organizations. What does this unauthorized use of bioresources means? When the multinational companies and organization, they don't have any right on that bioresource, but they are still using it. They are using it, they are selling it, they are doing everything which they don't have to because they don't have any right on that bioresources. That is what we call biopiracy. This occurs without compensating the countries and people who rightfully own those resources. So, when it happens, when we are using, we are not we don't have right on those bioresource of that country or that people, but still we are using it and we are not compensating, we are not giving any kind of money, label, anything to that country or that person, if it is belong to some people or all, or what is not rightful, like it is not right, and this is what we call biopiracy. So this is a figure showing biopiracy. So background of biopiracy, what is the global scenario, why it is the difference between this industrialized nation and developing and underdeveloped nations. So what actually happens, so these industrialized nations, they are very rich, okay, they are financially very rich, but they are very poor in their biodiversity and traditional knowledge. For example, in recent time, you guys have, this is not about biopiracy, but I'm just giving you an example so that you guys can relate. So nowadays we have seen that this this very big luxury company, this what we, I think this Gucci or something, I don't remember this fashion, it's a very big brand, and sorry, Prada, yeah, so Prada, so what they are doing, they have introduced this Kolhapuri chappal on one of their ramp walks, okay, one of their fashion show, and they are selling it at the amount of 2.5 lakhs, those normal chappals at 2.5 lakhs, and calling it their own fashion and all those things. But it, what it did, it created an outrage among the people, among the people who's actually this Kolhapuri chappal belongs to, this Maharashtrian, this, it is like famous from this Kolhapur, so this is, that's why we are call it as Kolhapuri chappal. So this is, it's like that only. So what happens in industrialized nations, they are financially rich, but they don't have this biodiversity and traditional knowledge. But in this developing or underdeveloped nations, the developing countries like in India, we are very rich in biodiversity, and we possess this very valuable traditional knowledge related to this bioresources. So what this financially rich company do, they use their financial power without giving us any compensation, without giving us any label or anything. They use our resources, they use our biodiversity and exploit that, and this is what we call biopiracy. So here we will see one example. So before that, let's see what is the issues and concerns. Traditional knowledge related to bioresources can be exploited by developed nations. So that, as I told, this traditional knowledge which is related to bioresources can be exploited by this developed nations. As I gave you this recent example, that this Kolhapuri chappal is like something that is made in Kolhapur and it is famous there. But this Prada, this big company, has used it, had launched it on their own name without giving any compensation or any kind of, label and anything to it, because this traditional knowledge help in developing modern applications. It saves time, efforts and cost in commercialization. So what is the issue and concern? First, this traditional knowledge, it is related to bioresource, it can be exploited by these developed nations. because this traditional knowledge help in developing this modern applications. We can use this traditional knowledge in making something very modern. And because we know that we in India we have a lot of this ayurveda and all those things which can be used for like treating even very big disease and all those things, so we can use this traditional knowledge to apply and on a very modern application and all. What it does, it saves time, efforts and cost in commercialization. So now what are the steps taken to prevent biopiracy? So there are some preventive measures that has been taken. Some nations are creating laws to address these issues. Issues includes injustice, inadequate compensation and benefit sharing between developed and developing countries. Because if you are using our traditional knowledge, you have to give the compensation, you have to share the benefit with them. But if they are not doing so, so this many nations have made some laws to address this issue, which the issues which include injustice, inadequate compensation and benefit sharing. This all is to prevent exploitation of their bioresources and traditional knowledge. What with these preventive measures will do, it will prevent the exploitation of the bioresources and traditional knowledges of those developing countries. So let's talk about some case study. The examples, as I was telling, examples which bio for bioresources, because we are talking about biopiracy, not other things. One of the example I gave just to make you guys more, you know, so you guys can relate. Here, biopiracy means when we exploit this bioresource things and all. So we will see the case study and then we will get to know it more. We will understand it more. So, case study first is basmati rice. So, basmati is a unique rice variety known for its aroma and flavor. 27 documented varieties have been grown in India. Basmati rice have been cultivated for centuries in India. It is its reference has been given in ancient text, folklore, art, and even poetries. So, basmati rice, it's a very unique rice variety. Why? Because it has some special aroma and flavor. Even we have also listened in that at home. Okay, this is basmati rice. This is very, it has a very nice aroma, it smell. It has very nice flavor and all those things. India in India, we have 27 varieties of basmati rice which have been documented and we are growing in India. Okay, this is a documented one I am talking about, 27. Now, this basmati rice have been cultivated in India for centuries, not like 10 years before, 20 years before, and all those things. We are growing this basmati rice in India. We are cultivating this rice in India for centuries. How we can say that? Because the reference of this basmati rice has been given in a lot of Asian text, folklores, and even poetries. So here's the pictorial representation. So now, what happened? In 1997, okay, so here I want to just tell you, in this chapter there are 2 years which is important, and both is same 1997 only, because the first transgenic cow which has been made, that Rosie, that's also developed in 1997, and this basmati rice biopiracy case, this also happened in 1997. So for your reference, if you guys can relate and learn it. So yeah. So what happened in 1997? An American company was granted a patent by the US Patent and Trademark Office. So what did patent this American company got from US Patent and Trademark Office? This patent covered basmati rice lines and grains. So what they did actually, how they got this patent? So what they did, I'll show you. What was the issue? So the company's new variety was derived from Indian farmers' varieties only. and this is like this new variety have been derived from this Indian farmers varieties only, but because now they got patent, so this allowed the company to restrict others from selling basmati rice. So even this basmati rice is something that have we Indians, the Indian farmers have been cultivated for centuries. This company has got this patent, and now they are saying that okay, so this is something we have discovered, and this company got the patent. And when you get the patent, then what happened? So other can't accept that company, other can't sell that basmati rice. Okay. So, and actually, why it was this? They were saying it was an invention or novelty. What they did actually, they took the Indian basmati, okay, and what they did, they crossed it with semi-dwarf variety, and the thing which is came out, the new, they said okay, this is an invention or a novelty, and now we have this right on this basmati rice, and now only we can sell this basmati rice, and others can't sell because now we have the patent. But that was wrong. What was the consequence of that? The patent covers similar products, okay, and this could limit other basmati rice sellers. Now they can't sell their own basmati rice because the patent covers the similar products also. But that's not the thing. That the thing they had made, that was a similar product, because what they did, they take a Indian basmati and then cross it with the semi-dwarf variety. But it was no new invention or novelty, but they did this, and this patent covers similar products, and this limit the other basmati rice sellers. So, what other examples we will talk about? There are ongoing efforts to patent products based on Indian traditional herbal medicines. Okay, so now people are trying to patent this Indian herbal traditional medicines like turmeric. We know that turmeric and neem are very beneficial to human. There was some legislative response. The Indian Parliament has recently cleared the second amendment of the Indian Patents Bill. It takes such issues into consideration. It includes patents terms, emergency provisions, and research development initiatives. So, as I told, the first example we have case study, we have talked about basmati rice, and now what we are doing that we are trying to patent products which are based on Indian traditional herbal medicines like turmeric and neem. Okay, so what was the response of the legislature, and what is like Indian government is doing in this? So Indian Parliament has recently cleared second amendment of the Indian Patents Bill, and it has taken consideration into these issues, and it includes the patent terms, emergency provision, and research and development initiatives. So now, what we have studied in today's class, what was the summary of today's class? First, we talked about the transgenic animals. What are the transgenic animals? They are the engineered, they are genetically modified, and with the foreign DNA, primarily for research or many biomedical application, which we have studied, to study for disease, to study the normal physiological and development thing, and for the biological product, vaccine safety, toxicity safety, and for all those things. As I said, they are used to study normal biological functions and serve as model for human diseases. Then transgenic animals are able to produce many valuable biological products for medicine and are used in vaccine and chemical safety testing, which we have studied. Then we have talked about the ethical concern which arise from this genetic modification, which can include ecological risk, and therefore there is a need for regulatory oversight. For example, in India we have India's GAAC. Then we talked about biopiracy. What is biopiracy? It is the unauthorized exploitation of traditional knowledge and bioresources is a significant issue. Example, basmati rice patent. Laws and vigilance are crucial to protect a nation's biodiversity and traditional knowledge from exploitation. Means we need to have some law and vigilance so that we are able to protect our nation's biodiversity and traditional knowledge. So this is the summary of today's class. So next is NCERT based question we will solve. So the first question asks us, What is molecular farming and provide an example? What is molecular farming and provide an example? Let's see what is molecular farming. So, it is the process where transgenic animals are used to produce useful biological products. So, even we have studied it in the necessity for this transgenic animal, that we need this transgenic animal so that they can produce some useful biological products. These products can include any medicine or human proteins which is beneficial to human. For example, we have studied about first transgenic cow, Rosie. What Rosie can do, it can produce milk which is enriched in human alpha lactobumin. And what this alpha lactobumin is doing, so this is very beneficial for the infant. It is providing more nutrition, a more balanced nutrition. And yeah, so this transgenic cow is able to produce that. Second question says, there is a controversy related to the patent granted on basmati rice. What was the core issue and why did it spark public anger in India? So we have studied about this case study of basmati rice. So they are saying ki what was the controversy which is related to this patent granted on basmati rice, and why, what was a core reason so that have sparked this public anger in India, because they are using our biodiversity, bioresources. Let's see. The core issue was this American company obtained a patent right on new variety of basmati rice. Origin of new variety was what? This new variety was actually derived from existing Indian farmers basmati varieties. Means what they did, they took this Indian farmers basmati rice, cross it with the semi-dwarf variety, and said that this is a new variety and now we have a patent over this. That this is some novel invention and novelty in they have novelty in this, and now they have only right for this. So it was, as I said, was created by crossing them with semi-dwarf variety. The reason for public anger? It was perceived as an attempt to patent and commercially control a traditional, long identified, and culturally significant Indian agriculture resource. This patent could potentially restrict Indian farmers and sellers from cultivating and marketing basmati rice freely. Because now they are saying that we have patent on basmati, then it can cause problem for the other Indian farmers to cultivate and market basmati rice freely. So what are the keynotes for today's class? First, the transgenic animals have manipulated DNA primarily for research, and 95% are mice. Transgenic animals produce valuable biological products, what we call also molecular farming, and aid in vaccine and chemical safety testing, which we have studied. Organizations like India's GEAC regulate GM research and product safety. Biopiracy is the unauthorized exploitation of traditional knowledge and bioresources. Nations are implementing laws to protect their bioresources and traditional knowledge. So there are some interesting questions that you guys gonna solve yourselves. So first question says, what defines a transgenic animal and why are mice the most common type of transgenic animal produced? Why mice is using as one of the most common transgenic animal? Why not other? You guys have to find the answer. Second question says, beyond research, identify and explain two practical benefit of transgenic animal for human well-being. So they are saying, beside this research work, beside what we are saying that we need to study a disease, we need to say study the normal physiology and development, why the other tell us some practical benefits of this transgenic animals. And there is also hint given to you guys that you guys can think about different products and safety testing. Question 3 says, what is the purpose of the Genetic Engineering Approval Committee GAAC in India? How does its role reflect the ethical considerations surrounding GM research? And why there is need of this GAAC in India, and how they are, how they are how their role is, like, how they're regulating this GM research and all those things by taking this ethical consideration into the factor. Last question is like, what actions are some nations, including India, taking to prevent the unauthorized exploitation of their bioresources and traditional knowledge? Means different knowledges which we talk even about legislative thing, that that second bill and all those things, amendment of second bill of Indian patent and all those things. What are the different actions India is taking to prevent this unauthorized exploitation of bioresources and traditional knowledge? Thank you. We will practice the DPP question. So, today's DPP questions, daily practice questions. So, question number first ask us, which of the following is the primary reason for creating transgenic animals that produce human proteins like alpha 1 antitrypsin? The question says, is asking us the major primary role or reason for creating transgenic animals which are producing human proteins like alpha 1 antitrypsin, all those things, all those proteins. So first is to study gene regulation, to reduce the cost of biological products, to test vaccine safety, or to improve nutritional value of animal milk. Why this alpha 1 antitrypsin is used? Okay, so the answer is, so the primary reason for creating transgenic animal that produce human protein. So to study the gene regulation, no, it is not like we have to study the gene regulation. That's why we are producing this alpha 1 trypsin. This is the biological product and it is helping us to reduce the cost of the biological products. Okay. Save and next. Then transgenic mice are frequently used to test the safety of polio vaccine. What is the potential benefit of this approach? Means transgenic mice are usually more oftenly is used to test the safety of polio vaccine. What is the potential benefit of this approach? Why we are using this? Because first option says mice are more affordable than monkeys. Okay, no. Mice can replace the use of monkey for vaccine safety testing. Okay, then mice have a longer lifespan for observing effects, or mice are immune to polio naturally. So the answer is second, that mice can replace the use of monkeys for vaccine safety testing. The third question asks us, which applications of transgenic animals involves introducing genes that make them more sensitive to toxic substance? So they are asking us ki which application. We have studied a lot of application of this transgenic animal. So they are asking which application of this transgenic animals involves in which we introduce gene that make them sensitive to the toxic substance. So we have studied it. So why it has been like used? First, study of disease model. No, in disease model we will insert gene or something that is going to cause that disease, not we are making it sensitive to toxic substance. Biological product production. No, in this we will insert some genes which is going to make a specific product for that particular protein or thing. Then chemical safety testing or vaccine safety testing. Vaccine safety testing we do to test the vaccine safety, but chemical safety testing or drug safety testing, in this what we will do, we will make this transgenic animal more sensitive so that we can see this chemical safety testing. Then what was the significant about the transgenic cow named Rosie? That what was very different and important about transgenic cow Rosie? So she was resistant to emphysema. Okay, then she produced milk with human alpha lactalbumin. Then she was the first animal used for toxicity testing, or she served as a model for Alzheimer disease. No, she didn't do that. She was used to produce this biological product. She produced milk with human alpha lactalbumin. Then last question asks us, which human disease has been studied using transgenic animal models? The question is says, this we have studied which among these human disease we have studied using transgenic animal models? Is it diabetes? Is it Alzheimer? It is tuberculosis, or is it malaria? So the answer is Alzheimer. This is a neurological disease, and there was some dysfunction in one of the gene that all those things. So we can use this transgenic animal model to study about Alzheimer. So this was all about today's class. So we will meet in tomorrow's class.